Good morning, everyone. I am Dr. Rituparna Shaha, Assistant Professor in the Department of Life Sciences, School of Biological and Forensic Sciences at Christu Jayanti Deemed to be University, Bengaluru, Karnataka. Today, I will be discussing a fascinating topic: centrifugation, introduction, and types of rotors. Centrifugation is a fundamental technique widely used in biological sciences, especially in biochemistry, molecular biology, and biotechnology. It plays a crucial role in separating cellular components and macromolecules based on their physical properties. Let us begin with an introduction to centrifugation. Biological centrifugation is a process that uses centrifugal force to separate and purify mixtures of biological particles suspended in a liquid medium. This technique is essential for isolating and analyzing cells, subcellular components like nuclei and mitochondria, and macromolecules such as proteins and nucleic acids. There are two types of centrifugation: analytical centrifugation, which is used to study purified macromolecules and their properties, and preparative centrifugation, which is used to separate and isolate biological materials for further use. Thus, centrifugation is both an analytical as well as a preparative tool in biological research. Now, let us understand the basic principles of sedimentation, which forms the foundation of centrifugation. When particles are subjected to centrifugal force, their rate of sedimentation depends on several factors. First, density. Denser particles sediment faster. Second, mass. Larger and heavier particles move more quickly in a centrifugal field. Third, the density of the medium. If the surrounding medium is dense, particle movement slows down. Fourth, the frictional coefficient. Particles with higher frictional resistance move more slowly. Finally, the centrifugal force that is applied. Greater force results in faster sedimentation. An important concept to remember is that sedimentation stops when the density of the particle equals the density of the surrounding medium. This is the basis for density gradient centrifugation. Centrifuges can be classified based on several characteristics. One key factor is the maximum speed at which samples can be spun. Higher speeds generate greater centrifugal force. Another important feature is whether the centrifuge operates under a vacuum, which reduces air resistance and allows higher speeds. Temperature control is also crucial. Some centrifuges are refrigerated, allowing sensitive biological samples to be processed without degradation. Additionally, centrifuges differ in their sample capacity, including the number and volume of tubes they can accommodate. There are several types of centrifuges available for different applications. These include low-speed preparative centrifuges, used for routine separation of cells. High-speed refrigerated centrifuges, used for subcellular components. Analytical ultracentrifuges, used for studying macromolecules. Preparative ultracentrifuges, used for isolating proteins and nucleic acids. Clinical centrifuges, commonly used in diagnostic laboratories. And microcentrifuges, which are used for small volume samples. Each type is designed to meet specific experimental needs. Now, let us move on to types of rotors. which are critical components of centrifuges. The first type is the fixed angle rotor. In this rotor, the sample tubes are held at a fixed angle, typically between 20 to 45 degrees relative to the axis of rotation. This design allows particles to sediment along the side and bottom of the tube, forming a pellet. Fixed angle rotors are widely used because they allow rapid sedimentation. Since particles travel a shorter distance to the tube wall, separation occurs faster compared to other rotor types. They are particularly useful for pelleting cells, isolating subcellular fractions. However, the pellet forms along the side of the tube, which may sometimes make resuspension slightly challenging. The next type is the vertical tube rotor. In this rotor, the tubes are positioned parallel to the axis of rotation, meaning they remain vertical during centrifugation. This design is quite different from fixed angle rotors. Vertical tube rotors are mainly used for density gradient centrifugation. Since the distance for particle movement is minimal, separation occurs very quickly. They are especially useful for separating particles based on buoyant density, such as nucleic acids. However, they are not typically used for pelleting because particles do not accumulate at the bottom in the same way as in fixed angle rotors. Another important type is the swinging bucket rotor. In this rotor, the sample tubes are placed in buckets that swing out horizontally during centrifugation. At rest, the buckets hang vertically, but when spinning, they move to a horizontal position. Swinging bucket rotors are particularly useful for density gradient separations. Since the tubes become horizontal, particles sediment in a straight path, forming distinct layers. This makes them ideal for separating different cellular components, fractionating biomolecules, and the clear layering allows easy collection of separated fractions. However, these rotors generally take longer time compared to fixed angle rotors. Thus, to summarize, centrifugation is a powerful and versatile technique used for the separation and analysis of biological materials. The efficiency of centrifugation depends on particle properties such as size and density, the centrifugal force applied, and the type of rotor used. Different rotors, like the fixed angle, vertical, and swinging bucket, serve different experimental purposes. Understanding these principles helps in selecting the appropriate centrifugation method for specific biological applications. Thank you for your attention. I hope this session has provided you with a clear understanding of centrifugation and the different types of rotors used in laboratory practice.